Welcome. I hope everybody can hear us, or at least me anyway, for now. If you can't hear it, please let us know. I'm gonna everybody here. Yeah, Eric just messaged. Okay, perfect, great. Welcome everybody. Good afternoon. Thank you for joining us. My name is Carolina Corlin, and I'm the Director of Marketing. I will be the moderator for today's webinar. Of course, we will be talking about PCR, antigen, and antibody COVID-19 tests, and essentially discussing what the various differences are between all those tests. Before we begin, I just want to do a bit of housekeeping. So as everyone is aware, everyone is on mute. This webinar is also being recorded, and so you will be receiving a recording of this via email with a link by the end of the day. There will be a Q&A period as well, and that will be towards the end. We will be going for about the full hour. We have about 40 minutes of presentation that Amanda will cover, and then from there we'll have a Q&A session. All right, so let's begin, and we will start off with our slides. So as we promised, our learning objectives for today are, the first one is to really review the testing methods and to highlight the major differences between PCR, antigen, and antibody testing. We will then move on to testing applications, and we will examine when and how each of these tests is helpful and useful to detect the virus. We'll also then move on to some testing limitations. So we'll review the limitations of each test and how that applies in testing scenarios. And then we'll finish up some testing implementation scenarios. So essentially going over how we can implement the various scenarios to help reopen the economy. So our speaker for today is Dr. Amanda Naaum. I hope I pronounced your name correctly, Amanda, or your last name anyway. Just a little bit about Amanda. She has about over 10 years of experience in molecular assay development for species identification in both academic and startup industry settings. Amanda does hold a PhD from the University of Guelph, and she has been awarded various sorts of awards, including the IUFOST Young Scientist Award in 2016, as well as the Commonwealth Rutherford Fellowship in 2018 in recognition of her research and innovation achievements in point of need food species authenticity testing. Sorry, that is a mouthful. She now extends this expertise to applications for food and animal safety, environmental monitoring, and human diagnostics at PBI as a co-founder and scientific liaison. And our co-host, Dr. Eric Hoskins, is with us as well, and he is a medical director with a PhD in public health and epidemiology from Oxford. An Order of Canada recipient, Eric has worked in healthcare systems in Canada around the world. He was Ontario's Minister of Economic Development and Trade from 2013 to 2014, and the Minister of Health from 2014 to 2018. Most recently, Eric was chair of the Federal Advisory Council on the implementation of National Pharmacare and has been advising the private sector on COVID-19 since the pandemic began. Eric brings his knowledge of healthcare and innovation to his role as board director at Precision Biomonitoring. All right, so a little bit about us. Precision Biomonitoring, or sometimes we call ourselves PBI for short, was founded in 2016 by a team of scientists from the University of Guelph's Biodiversity Institute of Ontario. Our very own triple-lock technology delivers on-site eDNA surveillance and human diagnostics test solutions. We provide detection tools for various scenarios ranging from laboratories to rapid, accurate on-site organisms and virus detection tests, including COVID-19. So to support the pandemic response, we offer industry-leading test solutions approved by Health Canada. Our first solution is our lab solution, which is the shelf-stable PCR COVID-19 test plates for lab settings, and this is of course made in Canada, and our mobile solution, which is a rapid, accurate, point of need on-site PCR COVID-19 test. And with that, I will ask Amanda to take over, and she will be continuing for the rest of the period until we are done for Q&A. And of course, our Q&A, you can see there's a chat box and a question box, so you can feel free to write all your questions in the question box or the chat box, and I will then read them out towards the end of the webinar. I hope everyone enjoys.
Okay, thanks, Carolina. So our first learning objective is around the differences between PCR antigen and antibody COVID testing. So the next few slides kind of just look at a high-level technical overview of what these different tests target and how they work. Can you go to the next slide? Great. So for PCR testing, PCR stands for polymerase chain reaction. It's a molecular test method that's used to analyze a short sequence of DNA, basically to determine presence or absence, in the case of SARS-CoV-2 testing anyway, of a specific target. So that target is amplified using a series of heating and cooling steps to make millions of copies, and then the test then can be used to detect that DNA. So there's various ways of actually visualizing what's happening there. In real-time PCR, sometimes referred to as qPCR, that actually analyzes the presence of those target sequences in real time with fluorescence monitoring. And for RNA, RT, or reverse transcription PCR, is used. So for these tests, you first convert ribonucleic acid, or RNA, into deoxyribonucleic acid, or DNA, with the reverse transcription enzyme, and then pretty much goes through the basic processes of heating and cooling that you would for typical PCR to be able to detect that target from a specific organism. So this method can sense even lower levels of genetic material, and they're very highly sensitive tests. These can be used across a wide range of applications, which include organism ID, which is how a lot of the bacteria or virus detection would work. It's also used in DNA fingerprinting and diagnosis of genetic disorders, amongst other things. Basically for any purposes where you're looking for a small amount of information or genetic material, and you need to make more of it to be able to detect it more easily. So next slide. So for COVID-19 in particular, PCR remains the gold standard to diagnose a COVID-19 infection. This method is not a new one. It's been used for diagnostics across a lot of different targets for a long period of time. These types of tests are designed to detect the genetic material or nucleic acid, as we discussed in the last slide, present inside a SARS-CoV-2 virus particle. So viruses, as part of their genome, have either RNA or DNA. In the case of COVID-19, it's an RNA virus, and so we're looking for RNA from SARS-CoV-2 in these tests. So this type of testing can be performed in a couple of different facilities. Either that can be a traditional laboratory test where samples are collected and sent out to a facility, like a centralized sort of approach. There's also point of need or mobile laboratory settings as well, depending on the equipment that you have and the throughput that you need. Both of those are possible applications. And because this test is sort of the standard or reference type of test that's used, this generally can be used without additional confirmatory tests, which is nice. So you've got one test and you've got your information through which you can make a diagnosis. Antigen testing is, not surprisingly, targeting antigens, which are composed of proteins, peptides, and polysaccharides, and essentially is a substance that induces the immune system to produce antibodies against it. So basically, a virus, in this case in relation to COVID-19, but others as well, contain distinct sites on its surface. So these antigens will have these epitopes that then connect in with an antibody that's generated to recognize and interact with it. So these antigen binding sites or paratopes are what triggers immune responses. Any portion of bacteria or viruses with either surface proteins, capsules, toxins, etc., can potentially serve as antigens. And an antigen test would then detect one or more of these specific proteins, making use of these really specific interactions between antibodies and epitopes. So some common antigen tests are for streptococcus, for influenza, for malaria, and others. For COVID-19 in particular, antigen tests have been designed to detect certain proteins on the SARS-CoV-2 virus. In general, antigen tests are highly specific, but are typically less sensitive than molecular tests like PCR. They can be also, though, generally produced at a lower cost than PCR tests and have a simpler design. So this means that you could potentially scale to millions of individuals per day, which is more difficult to do with PCR testing. Antigen tests are often referred to as rapid, and they would provide results in less than an hour. And most of the currently authorized tests are related to antigen testing for COVID-19 are point of care and use lateral flow technology. And finally, for antibody testing, is a whole other essentially protein that's being detected. So antibodies are created by your body's immune system after infection and or vaccination. You may take weeks to start making these antibodies post actual exposure to a virus. And these antibodies then help your body fight viral infections, potentially can be more easily made when you're infected with the same virus again. For antibody testing, or commonly referred to as serology tests, these look for specific antibodies to specific viruses in your blood to determine if you've had a past infection. And for COVID-19 in particular, again, the antibody test is looking for COVID-19 specific antibodies in the blood. This is the least commonly used testing method for COVID-19 in that it shouldn't really be used to diagnose a current infection with the virus that causes COVID-19. And that's just because, as we discussed in the previous slide, it can take some time for these antibodies to build up. So it would not necessarily show if you have a current infection, but it would tell you if you had one in the past. And since COVID-19 is a relatively new disease, we're not sure yet what protection antibodies actually give you against future infections and for how long. So there's ongoing research into this question. So in summary here, for PCR tests, we're looking for pieces of genetic material from SARS-CoV-2. It tells you if the person has an active infection, and in most cases, this involves a nasopharyngeal or nasal swab taken by a healthcare provider and then tested with PCR, either at an external laboratory or possibly while you wait with a mobile laboratory option. Sometimes for some tests, oral swabs and saliva can potentially be acceptable sample types. For antigen testing, we're looking for proteins that make up the SARS-CoV-2 virus. It also tells you if the person has an active infection. In most cases, it's a nasopharyngeal or nasal swab again, but these tests can be run while you wait. Occasionally, there may be a sample sent to a lab for testing. And then finally, antibody tests are looking for antibodies made by your immune system to indicate some immune response against SARS-CoV-2, determining if the person had had a past infection. And these are usually done with blood samples. So our second learning objective is around how do you apply these different tests. And so there's a few slides around how they might be used and applied. So in PCR testing, normally this is used to determine active infection regardless of symptoms. So because of where the sensitivity is for this test, you may be able to detect in asymptomatic individuals. It can also identify people who are contagious. In communities where transmission rates are low and mitigation efforts are effective, these are the types of settings in which PCR test applications are best used. This is a gold standard test for doing this type of identification. So it identifies people who are and who are not infected, so kind of a presence-absence. And those results can be available within potentially a couple of hours if you're analyzed on site, and then a few days if sent to a lab. There are some on-site PCR lab testing solutions available through Precision Biomonitoring, which we're going to talk a very little bit about towards the end of the presentation. For antigen test applications, this is, again, is a point of care test. So this is quickly used to determine who has an active infection. It is a less expensive approach to do that than PCR. And it does perform best, though, 5 to 7 days or so after symptom onset on average. You could have on-site test results in less than an hour with this type of testing. They tend to be highly specific, but are generally less sensitive than PCR tests. And there can be, because of that reduction in sensitivity, an increased risk of false negative results. So a lot of the standard testing protocols, procedures out there, as far as flow diagrams of how to do your testing and interpret results, will suggest a PCR test to confirm negative results. For antibodies, again, this is not used to diagnose current infection, but it can identify that there was an infection in the past, even if you're no longer carrying symptoms of the illness. This just determines that there has been COVID-19 in your system, and there can possibly be some opportunity to identify when that happened, depending on the types of antibodies that are being tested for. This may also potentially be applied to determine qualifications to actually donate plasma for treatment purposes. Antibody testing also helps public health and researchers track the percentage of infections that have happened in the past. It should not be used to determine return to work or schools or anything like that. And it may also be applied to measure vaccine success, so that you actually have started generating antibodies post-vaccination. And just to kind of summarize in terms of the progress of the illness and the tests that may be used to actually identify active infections, this graph shows on the x-axis at the bottom days since onset of symptoms starting at 0, and days since potential exposure or infection kind of towards the -5 there at the left end. On the y-axis is showing the viral load kind of as that infection makes its course. This is sort of on average, again, for both the testing as well as the viral load, but this gives a good overview of kind of what's happening through time. So at the initial onset of infection or exposure, there will be a little while where maybe no tests are going to necessarily pick anything up because the viral load is very, very low. But starting usually several days before the onset of symptoms, PCR can be used to identify an active infection of SARS-CoV-2. You'll see at the top there are PCR tests, and then there's an extension of digital tests as well as visual tests. Both of those refer to lateral flow testing or antigen testing. Some of these are based on a visual identification, where others employ a digital reader, which kind of extends the viability of that type of testing. So a little bit before a visual test might pick up, there might be enough viral load to pick up an active infection. With a visual test, digital tests may be able to actually see that a little bit before symptom onset. And then, as you can see, as the infection progresses, the viral load is decreasing over time until it is expelled from the body. So at that end stage as well, PCR may be able to pick up the presence of viral particles, where some of the other tests will no longer be able to do so. So in summary, as far as how to apply these different tests, every day of infection can be very impactful. Tests with higher sensitivity in theory allow then for earlier detection of the virus, potentially before then the onset of symptoms. PCR testing continues to be the most common type of test used and remains the reference test, but each of the tests has a place in the testing cycle, and depending on goals and implementation, there may be some that are more appropriate than others. So for PCR, you're going to get the most accurate results, and detection before symptom onset can potentially be performed on site, but it is the most expensive and slowest way to test. Antigen tests have point of care pretty much across the board, so this can be done quickly and cost effectively on a larger scale than PCR, but there is some lower sensitivity in comparison, and as a result of that, false negatives occur at increased rates comparatively. And antibody testing, again, doesn't do anything for current identification of infection, however, can be used to identify past infections and is a valuable research tool, as well as a tool for monitoring outbreak investigations. So our next few slides just talk about some of the limitations of these different tests. So we've spent most of this time talking about why each of them is great and where they can be used, but each of them also comes with some limitations. So for PCR testing, it does not determine past infections. Again, that's not something that PCR will say. It's only for the point in time where that specimen was collected. It also therefore then can't predict if you will remain negative. So it really is a snapshot in time. In some people, the virus can also be found by PCR in the nose and throat for several weeks, potentially after that person is no longer infectious. And it generally requires certain kind of swabs and reagents that may be in short supply. So I had mentioned there are some PCR tests that can be used with saliva or with oral swabs, but in general, they require a nasopharyngeal swab and transport media that can be problematic to obtain. For antigen testing, the best results are achieved with people who are symptomatic already. So, and in addition, it also, similar to PCR, will not help determine who had a past infection. It only identifies a current infection. There is some lower sensitivity with this test, so there may be false negatives compared to PCR. And in persons with symptoms or known exposure, therefore those negative tests can only be treated as a preliminary result, and it's suggested that they're confirmed with PCR testing as well. So you can do rapid screening, but there's potentially a secondary testing stage to have full confidence in the outcome and diagnosis. And for antibody testing, if you're actually looking again to find if you'd had a past infection, if you do it too close to when you are infected, you may get a negative result. So it's important to consider the timing of the testing, as with antigen testing and PCR as well. There can be low sensitivity and specificity results, and there may be some cross-reaction with other coronaviruses that are not SARS-CoV-2. So again, because this is generally used as a research or predictive tool, that needs to then be taken into account with an additional amount of epidemiological and clinical data to have that data be useful. At this time, we don't know how long and how much antibodies actually protect from repeat infection for COVID-19. And just an encouragement to continue to take steps to protect yourself and others, even if you had an antibody positive test as a result of that. So the next couple of slides just deal a little bit, we've used the term sensitivity a few times. So just defining that term in the context of a couple of other important terms. So for sensitivity, a test sensitivity comes down to a fraction of positive cases that the test correctly identifies as positive. So out of those known positives, those persons have actually got the disease, how frequently is the test that you're using also giving you a positive result? So for example, if you have a test with 80% sensitivity, the test would detect 80% of patients with the disease, but 20% of those patients would be not detected, and considered false negatives, essentially, by the test. So a highly sensitive test will have a low false negative rate, but if you don't also have high specificity, you run the risk of false positives. And sensitivity may also be referred to as true positive rate. For specificity, this is the fraction of negative cases that the test correctly identifies as negative. So sort of the inverse of sensitivity. And it mentions how frequently the test is negative, or measures, not mentions, excuse me, how frequently the test is negative when that person doesn't have the disease. So out of a group of people that do not actually have the condition, a test that has 80% specificity will basically show that 80% of people without the disease are negative, but show 20% still positive. And that would then be termed as a false positive with the test, and that false positivity rate as something that you'd want to avoid because of all the potential implications from that. So a highly specific test will have a low false positive rate. And again, you want to balance that out, though, with sensitivity, because you could then have a risk of false negatives if the test sensitivity is low. And again, specificity is then sometimes called true negative rate, conversely with sensitivity as the true positive rate. And then finally, for positive predictive value, this is a measure of how likely a positive test is a true positive rather than a false positive. And this is a little bit more complicated of a calculation and depends on how many people in the population have tested to have had the disease. And where there are very few people in the population that have had this, then there's higher chance for false positives. But where you have a system where many people in the population have had the disease, then there's a higher chance that a positive test is a true positive. So in general, to kind of summarize the last few slides, what do your positive test results and negative test results mean? Well, for PCR, a positive test result means the person tested has an active COVID-19 infection, that virus RNA is present in their system. And a negative test means the person was probably not infected at the time that their sample was collected, not necessarily that they don't have trace amounts or early in the infection or that they won't get sick, just that there is no detectable SARS-CoV-2 at the time of testing. An antigen test is similar to a PCR test in terms of positives. It means that that person has an active COVID-19 infection. There is virus present and detectable in that person's sample. And a negative antigen test means that there weren't any SARS-CoV-2 viral proteins, particles detected with the test. In persons with symptoms, a negative test doesn't necessarily rule out COVID-19, and quarantine is suggested with a confirmatory PCR test. So this is something to keep in mind as far as the usability of the data coming out of this and what confirmatory testing may need to be completed. For an antibody test, again, not for really detection of COVID-19, but a positive test would mean that you had been previously infected with COVID-19 and the immune system did develop antibodies to try to fight off the infection. A negative antibody test means that that person may not have had COVID-19 in the past. However, they could still have a current infection where the antibody test was conducted too soon to give a positive result. And again, it's not known how long these antibodies stick around at the moment for this particular virus. So it doesn't necessarily mean that you've never had COVID-19 either. In summary, for PCR tests versus antigen testing, sensitivity and specificity is important to consider in terms of developing and implementing any kind of test plan. PCR, very generally and across the board, kind of on average between these tests, PCR tests are going to be highly sensitive, especially traditional laboratory PCR tests and reference methods, and highly specific. Antigen tests, conversely, are still highly specific, but often are less sensitive than molecular test counterparts. It's important to remember all of these sensitivity and specificity information is generated by comparison to reference tests, which themselves have specificity and sensitivity values associated with them. And none of these tests are 100% accurate all the time, but this gives an idea, these values of sensitivity and specificity, of the confidence you may have in the results that you're obtaining. And the test performance can also vary based on the prevalence of the disease in the population being tested. Some other things that may affect the test accuracy include that you have the virus, but the swab is collected incorrectly or misses the virus from your nose or throat during the sample collection. The sample could potentially be contaminated with virus during collection or analysis, causing a false positive. The nasal or throat swab may not have been stored correctly. So whether that's temperature or media storage requirements, it's really important that the test is done on a viable and properly stored sample. And the chemicals used to extract the virus genetic material or actually do the testing of that material may not work correctly. So whether that's in the antigen testing or PCR as it's described here, there can be failures in the chemistry of the test itself. Most tests have some built-in controls to try to identify those failures, but they can still occur. And the last piece of information that we're going to be talking about is test implementations for PCR antigen and antibody options. So the first question is why implement a testing program? I think really the goal with these can vary, but in general, it's to create environments where people can continue with their lives, going to work or school safely, allow return of employees and students without reducing density or moving to shift work or otherwise adjusting the typical workplace and productivity. You can massively increase the scale of testing to do contact tracing and capture asymptomatic cases. And it also enables additional data to make effective decisions and do research on COVID-19, as well as future pathogens and potential future pandemics as well. So in terms of how to implement a testing program, this is definitely going to vary based on your goals of your actual program. But in general, you've outlined 4 steps that you want to definitely touch on pretty much regardless of your goals. So the first thing you want is creating the actual testing program, and that should take into account, as I said, the goals of what you want to achieve with it, what the end result is that you want to have, the throughput that you would potentially have, what time to results do you need or are you looking for, and incorporate that into a comprehensive plan that uses some type of a approved private testing kit that demonstrates some efficacy as compared to what's being used in public tests, so that there can be some confidence in the data and there's not as much comparison testing and downstream testing that would need to be done, and something that delivers rapid results, again, in line with the goals of the testing program. Step 2 is to actually then implement that testing. So whether that's at work sites or schools or wherever the sort of setup makes sense for your particular application. And there should be a testing program in place with regular intervals for all employees or other members of the specific community that's under the umbrella of the specific testing program. So from that testing program, which again should take into account clinical information, rates of infection in the population, likelihood of exposures, and again, how often do we want to have to send people to have a comparison test done, etc. So for step 3 is how do you then manage the results that are coming out of this testing? So you want to have a system that has anonymous and rapid results that are secure. A system for what to do when employees are testing positive, and a contact tracing system in place, either internally but also reporting to public health as well. And then for the last stage, for employees that do generate, or again other members of the test program community generate a positive test, then there may be some qualified contact tracing systems internally to support public authorities aggregating the data from both that person as well as close contacts and helping to manage contact tracing efforts. So in general, all of the protocols used should consider the risk level of the setting. You want to have a transmission reduction or elimination goal that is in line with that risk level that makes sense for the specific community or location. The testing strategy should also look at accomplishing transmission reduction goals, so that once those transmission goals are set, how then do you best, with all of the available options, meet those transmission goals, the reduction goals the best, and then have processes for acting on the testing results. And this is something really important, which is mentioned in the previous slide, but I'll reiterate here, is that you definitely want to have some idea of what to do when somebody tests positive, and a really clear protocol, so that the testing can be useful, can be acted on. So you'll look at test choice in terms of the accuracy of the test, the convenience for your particular goals, the time to results and cost, as well as throughput, who to test, how often to test them, and then there can be advanced strategies as well within that. So as far as precision biomonitoring products that are available, we're going to talk about two test solutions. The first is our laboratory testing solution, and this is our Made in Canada product. So it consists of a 96-well plate with lyophilized reagents. You'll see them as little beads, pre-aliquoted and dried into plates. So these can be stored at room temperature, everything thermostable, and essentially you add your RNA extract to these and go. It seriously reduces the number of steps and therefore opportunity for contamination, as well as the cost in terms of labor for doing these steps. The tests for our Health Canada submission in comparison to a reference test showed 100% sensitivity and specificity. It is a multiplex test. It targets 2 viral targets as well as an internal control to confirm where you have negative results, that result can be relied on and there wasn't a failure in the actual test itself. As I mentioned, everything is pre-mixed and ready to use, and can help minimize errors and contamination, as well as decreasing costs, because it takes a little bit less time to mix everything together and go. And it can be used in multiple thermocyclers, so basically anything that will fit the plate can be validated for performance with this. So overall, the goal with these is hopefully faster test results because of the reduction in numbers of steps and freezing, thawing, mixing steps that are required with traditional lab testing. On the mobile solution side, we also offer a solution that's designed for rapid on-site diagnostics. This is also Health Canada authorized. It is a laboratory test, but it is meant to be mobile. So it can be deployed in relatively small footprint with trained technicians and personnel. So there are fewer preparation steps here compared to what you would be doing in the laboratory, and also is a shorter test. It also entirely eliminates, as with our mobile option, the use of wet chemistry. So all of the tests are lyophilized again, freeze-dried into a thermostable format. They're in the tubes are shown there at the bottom picture on the right, and those have already been filled with sample. There is also showing on the top photo field extraction system to do RNA extraction rapidly without any complicated equipment. So in that photo at the top, you see everything that you require outside of cleaning supplies and PPE to actually run this test. So it is a very compact system. After extraction, the PCR test itself is about 1 hour. So it is relatively rapid. Depending on the throughput, that turnaround time could be a couple of hours only from the time the actual sample is collected. So fairly simple and rapid to implement for low throughput applications in mobile settings. Kind of a non-traditional lab approach. So for this mobile product, there's 3 tests. It's relatively straightforward. You're going to collect your sample with some type of nasal fluid. You're preparing the sample. So this is another close-up of that extraction system that I had mentioned, which inactivates the virus as well, and then prepare it for testing by adding it to the lyophilized reagents directly. And then you process to get results using RT-qPCR and a provided mobile thermocycler that's shown in that 3rd photo. The smartphone that's shown is what's used to actually control the device and also records the results. The results are also then recorded, if preferred, on a cloud system. So that means that if you're running multiple mobile laboratory locations, those results can be tracked in a centralized capacity. So the benefits of our mobile solution have really good sensitivity and specificity at 98 and 99% respectively. It's pretty rapid for 9 tests, which is the throughput of 1 instrument. You can get from sample to results in about 2 hours with minimal prep. So there's minimal steps and minimal equipment required. Everything is fully portable with this system. So everything, including the RNA extraction, is ready to use and thermostable. There's just those 3 simple steps that we went over. It's relatively intuitive as far as the app and interface to run and monitor the results in real time. And you have that cloud connectivity for easier remote access and sharing of data. Everything is pre-mixed and ready to use. No cold chain, as I mentioned. And this test is also a 3-target multiplex with 2 internal viral targets and 1 internal control, again, to test for reaction success. And our last kind of slide note here is a little bit of a case study on one of our customers. So New Gold is an intermediate mining company with operations in Rainy River and New Afton. And they were challenged at the beginning of the pandemic with shutdowns at Rainy River for 3 weeks, which delayed major projects, they were impacted by border issues for equipment. So really needed to have some kind of a solution to get back up and running. So for assisting them with their testing strategy, they were looking for something that was quick and accurate and that could provide peace of mind to employees to be able to return back to work safely and to be able to work in close conditions without issue. So we were able to provide the RT-qPCR mobile testing solution, and that was implemented at Rainy River. It's been executed after PBI provided training and support. This solution has enabled New Gold to steadily increase capacity. They are now back up to 100%. They are testing at the beginning of each shift, and they're now looking at implementing this in their newest mine in British Columbia.
Okay, great. Thank you, Amanda. So now we'll take questions. We'll take the next, a little bit under 15 minutes for Q&A. I'm going to read the questions that as they appeared in chronological order, starting with the very first one, I believe was by John. How do you adapt tests for new variants, or are they all detectable? Please elaborate. So either Amanda or Eric, please jump in.
Yeah, I can take this one. So our test, both versions of our test, I should say, our mobile test as well as the Canadian-made one, are not affected by the variants in that they will still give you positive identification with those variants. However, neither of them is designed to identify specific variants. That's sort of another test that would need to be done on them.
Okay, great. And we'll move on to the next one. I think the next one was from Christina. What are the challenges associated with qPCR for virus detection? Are enzymes-linked immunoabsorbent assays more specific? What are the pros and cons of both?
I will say that, I mean, ELISA is a little bit outside of the scope of this particular presentation. I know it can be used successfully as a diagnostic tool. In our experience and in sort of the availability of diagnostic testing, really qPCR is that gold standard that you're looking for. It is routinely used for diagnostics across a wide range of targets, way before this pandemic and COVID-19. I don't know that I have a comparison data percentage that I can give you, but there is a reason that, you know, PCR is the kind of go-to for this type of testing. There are always challenges with whatever opportunities, you know, you're looking at, as we've discussed, kind of pros and cons of these different methods. But qPCR really does remain, for a reason, that gold standard. And we can definitely maybe follow up with some specifics around ELISA. I'm hesitant to give you any numbers without anything kind of at my fingertips today.
Yeah, definitely. It looks like the next question was around swabs. I believe it's Israel who asked how long the swabs can wait before testing for antigen. So I'm wondering if that is a question around surrounding expiries in terms of swabs. I'm not quite sure.
I can take that. Amanda, I'm happy to take that one. I think obviously every swab has an expiry date, but it's probably referring to, once an individual is swabbed, how long can you safely wait before that has to be tested in the laboratory? Generally, it's recommended, regardless of that lag time or the wait time, it's recommended that you refrigerate the swab. But even without refrigeration and specific to COVID tests, you can safely wait up to 48 hours with that swab unrefrigerated, as long as it gets to the lab within that 48-hour window.
Great, thank you for that. Our next question is Mohammed. If you have a COVID vaccine, are the antibody tests, can the antibody test still determine if you develop antibodies or not?
Amanda, I'm happy to jump in on that one too. So, Eric, the answer is yes and no. So you can test positive after vaccination, assuming you develop a proper antibody response. There are some antibody tests, COVID antibody tests, where you can test positive with that antibody test, but it's not a guarantee. There are also it depends on how the antibody test itself was developed, and so you may test positive or you may not test positive. And also, it's important to recognize that there are different aspects to your immune response, right? There's not simply the development of antibodies. There's also the development of T cells. So it's more complicated then, and which is why CDC and other public health authorities are not recommending, for example, an antibody test after vaccination to confirm whether or not you are protected, because sometimes those antibody tests will test positive, but sometimes they won't, and that really has no direct bearing on whether you're protected.
Okay, great. Thank you for that, Eric. Next question comes from Michelle. Can you use a qPCR TaqMan assay to detect and identify variants?
So in theory, yes, and I think there are some commercial ones starting to appear to be able to do that. As far as I understand, there hasn't been anything authorized by Health Canada for diagnostic purposes for this yet. So the ones I know about are research use only at the moment. And of course, then you are limited to testing for portions of the variant. So each kind of variant of interest has many mutations, and so you are in a position to sort of choose the ones you want to interrogate. So it's a little bit more complicated than the straight kind of diagnostic yes or no that we're talking about with the PBI products, but it is possible to do.
And maybe just to add, Amanda, as well, to give reassurance, virtually every PCR test that's out there will detect the variant as positive, because the portion of the genome that is targeted by PCR tests is one which is stable and not impacted by mutation, like in a substantial way. There's it's the N501Y, which is the gene mutation that occurs actually for all three of the major variants, if we can call the Brazilian, South African, and UK variants that. So there's, as Amanda has been saying, there's two steps. We can all be reassured that PCR tests will still be positive if a person is infected with COVID, even if that's a COVID variant. The PCR test will result in a positive. And then there's a secondary test that's required, which focuses on that specific gene mutation, as I mentioned, the N501Y, that will tell you which variant it is.
Okay, great. Thank you. So we have another question. There's lots of them, hoping we can get through all of them. So this one's from Sheila. She's asking if we can comment on the implications of using diagnostic tests versus screening tests.
Eric, if you're...
I didn't hear the first part of that question, sorry.
Sorry, I'll repeat. Can you comment on the implications of using diagnostic tests versus screening tests, or as screening tests, sorry?
Oh, well, you know, in a perfect world, a diagnostic test would be a better screening test than, say, an antigen test, because it's more sensitive, similar specificity, but it's likely to pick up more cases in the community. And so so what you want to avoid, obviously, is false negatives. And so, however, the PCR tests are significantly more expensive than the antigen tests. So typically, the main reason for antigen used for screening is its ease of use from a technical perspective and its cost. But there are limitations, which most specifically are the lower sensitivity. So you can expect to miss what are true positive cases of COVID.
Right. Great. The next one is from John. This again asks about details on detecting variants. I know we covered that a little bit. I don't know if there's anything else that we want to add to that specifically.
Maybe just to, there's another question in the chat which is related that says, does the virus capsule remain constant with the variant and the genetic material change?
Oh yeah, by Bill, yes.
Variant, it's that very narrow specific part of the genome, that gene mutation that I mentioned, the N501Y. So the capsule is unchanged so far.
Thank you. So the next one is from Christina. As the vaccination increases, are you concerned about false positives? Is it a possibility?
So no, it's not. So you don't test positive, like a true positive, after vaccination with a PCR test. For a couple of reasons. If we look at the mRNA vaccines like Pfizer and Moderna, for example, the mRNA only lasts in the body really for a matter of hours, maybe a couple of days maximum. And when the mRNA, when it catalyzes the production of the protein, that small amount of the spike protein within the cells, there's no migration of that protein to the nasal passages. So when you swab for a PCR test, there's no chance that you'll pick up any of those spike protein fragments because they simply don't travel to the nasopharyngeal area. So the short answer is, if you test positive after vaccination using a PCR test, either you have a real infection that you might have got immediately prior to or immediately after vaccination, or the vaccine didn't work, or it's a false positive.
Okay, great. Thanks, Eric. All right, so our next question is from Jonathan. He says, the studies for PCR tests showed that sensitivity peaks 7 days after exposure. It seems that if the median of symptom onset is 5 days after exposure, PCR isn't actually very good at picking up pre-symptomatic individuals. Can you confirm the point you made that PCR can pick up infection prior to becoming symptomatic?
Go ahead, Amanda.
Oh, I just wanted to clarify. So on that graph, what that's showing over time is a viral load. And so definitely there are obviously peaks and then valley to kind of how that infection is progressing. What that is really meant to show is that just strictly from the sensitivity of the method, there is no real opportunity for antigen tests to be able to detect in that early zone. Certainly there's a lower level of virus present, but it should be above the detection limits of most of the PCR tests, just depending, again, on average infection rates and number of days across individuals. So it wasn't meant as like definitely from 5 to 7 days is really an average across them, and that there is fluctuations in viral load where on that first few days other test methods just are not capable of picking it up. So
yeah, and I'll just add to that, even with PCR, there have been many studies done, and from the time of exposure, right? So this isn't about symptoms, but from the time that you might have been exposed to another person with COVID, 2 days after that exposure, 98% of the time, you're still going to test negative by PCR. 5 days after exposure, again, this has nothing to do with symptoms at this point. 5 days after exposure, it's 50% will be picked up by testing, and then after 5 days, only 10% will be missed, so 90% will be picked up. So the individual that asked the question, that's a really, really good and important question, and that's why, as a public health doctor, for example, I advise, like, people, if they get exposed, I advise them to self-isolate, to wait 2 or 3 days before getting tested, or because then they're more likely to test positive if it was a true exposure, or if they feel compelled to test immediately. I tell them, even if negative, they should retest in several days following that.
Great. That's great. Okay, so we're gonna skip the other question that we've kind of covered earlier. I think the next question after that is sorry, I think it's David. It's Ritu. I apologize. How does the testing change with respect to variants?
I think we kind of covered that, didn't we, Amanda?
Yeah.
Go ahead.
And it's sort of it's kind of a secondary test if you want to identify the specific variant. And as Eric clarified in that initial question, the tests we're talking about for actual diagnosis, they're just going to still tell you positive. If you want to identify specific variants, you would need another test to identify which one, or if it's a variant of concern. And just to reiterate on that, that's something that is evaluated as part of the Health Canada requirements for authorization. Post-market is continued to be evaluated, that the presence of these new mutations does not affect the test. So it's something that, as they're identified, go back and check based on the test design, is that going to impact performance or not? So it's not a guess. We look at it, and it's on an ongoing basis for both of the tests that we sell, and that will be across the board for other authorized tests as well.
Okay, great. Thanks, Amanda. The next question is from David. Are longitudinal studies of previously infected and or vaccinated hosts been or are planned? We have a large pool of infected people to draw from.
Yeah, so the answer to both parts of that question is yes. So there are many longitudinal tests, or rather trials, observational trials of both of individuals who have been infected with COVID, as well as looking at individuals who have been vaccinated. And both are obviously important. A lot of the information on long COVID is coming from those observational studies, but also with regards to post-vaccination, it's really important to determine how long the protection as measured by antibody and T cell response, how long that protection will last. So the answer is yes, many studies.
Great, thank you. Next one, we're almost done through the questions. I apologize, we are running a bit over time, but I do want to try and get most of these answered. This one is a product question, Amanda. It is, what is the max volume per well?
So we don't work on max volumes because we have a specific dilution required for the beads to maintain the concentration of those reagents. So it's 20 microliter reaction for both versions of the test, actually, just coincidentally. But you add 20 microliters of RNA straight to the reagent, and that's it. Mix and go.
Okay, perfect. Thank you, Amanda. Any other product-specific questions we can probably handle individually through our reps? So there's one about how long the order would take for pre-filled plates, and we can connect with that with Samantha, with you on a later note. There is a question about the genes. So what are the 3 genes? Which genes are targeted in the internal control, and which are the targets? Amanda, it's probably for you.
So it's actually different for both of the tests we offer, and it will be different across other PCR tests as well. Our triple lock test targets UTR and E gene, and the control is an RNase P human gene. The mobile version of the test is spike and ORF1AB, and the control is actually a synthetic spiked in control that goes through the full extraction step, just as an additional measure with that mobile field prep to make sure everything went through as intended.
Okay, great. The next question is from Vasco. What do you do if one of the viral genes tests positive and the other one is negative?
So I am going to say a very basic answer to that, and then say you will consult our instructions for use for both tests, because it's a little bit different for both. Essentially, in general, that can mean 2 things. It could mean a low level infection where only one's being picked up. It could be alternatively that they're actually, and this is part of the reason we have two gene markers, that there is some mutation that is causing a failure in one gene. So I think for both products, we recommend a retest and to handle those results depending on the outcome of the secondary confirmatory test. So you'd run the same test again in-house, see if that result is confirmed, and then make your assessment on the diagnostic positive or negative, and or necessary reporting to public health from there.
Okay, great. This is more of a follow-up as well. This is with 2 target detections, how would you interpret the variants?
There isn't any I just want to clarify, there is no variant information that you're going to get from the test that we've discussed today anyway. And again, that's true for all of the current SARS-CoV-2 just diagnostic tests. So you won't get any variant information. What we know is that you shouldn't have any change in results. It should just still tell you positive, whether you've got wild type or one of the mutations present. But you can then send that sample, if you have a positive, for additional testing to determine if it's a variant of concern.
Okay, great. Thank you. There's some specific product questions which we'll answer privately. There's one here. What are the chances of mutations that can result in a positive case being undetected by PCR testing? This is in regards to the case that occurred in France.
Yeah, and that was very recent. That report, I think, was just a week ago, that there were a number of cases in France where they initially were undetected using the traditional PCR tests. Subsequently, they were deemed to be positive, either through antibody testing or through, I think they did a bronchial wash. There was like a more substantial sampling that was done. So you may remember earlier on when I was talking about how all the PCR tests can pick up the variants. I think my very last words were like "so far." So that is a risk depending on where the mutations occur. And I believe the mutations in the French example were on the spike protein. But that is, it's very early days, right? So I would kind of hold my breath on that until we have more information of whether this is truly a variant which can evade at least some of the PCR tests or not. I think we need to wait and see if that happens. This is the first example that I think there might have been an example in Finland as well earlier, but this is obviously a very rare occurrence.
Great, thank you. Okay, I think we're going to probably start wrapping up. We'll take one more question, and then what we'll do is we'll leave an email at the end where you can reach out to us if you do have any other questions or are requesting product information. So here's the last one. Some antigen tests I've seen have indicated high sensitivity, somewhere around 93%, and high specificities around 91 to 99%. Are they playing with the numbers? How are we to determine the best antigen tests?
Well, yeah, I'm happy to start on that. Like, my feeling on the antigen tests is that it's not that they're overblown, but these are studies done in a laboratory environment, and so there are a number of antigen tests that have claimed in the lab concurrence or say sensitivities in the low to mid to high 90s, but then subsequently researchers have done real life out in the community testing using these same tests, and they found that in fact the sensitivity can be as low as 70 or 75%. So I think, and similarly with PCR, it's only as sensitive as the capability of the person doing the swab sampling as well. If you get a bad swab sample, you're going to miss the virus. But certainly with antigen tests, those are in perfect laboratory conditions. And so I would frankly knock like 5 or 10 or 15% off that sensitivity.
Great. Anything else, Amanda, to add to that, or?
No, that's, I think, right on.
Great. Awesome.
Just important to consider.
Yeah, absolutely. Okay, well, we're going to wrap it up now. It is 10 after 1. I want to encourage anybody who has any other outstanding questions to please contact us at info@precisionbiomonitoring.com. You can also connect with us via social media. And then, of course, we will be hosting other free webinars. So sign up to receive some upcoming notifications on our website at precisionbiomonitoring.com. We thank you very much for sharing your lunch hour with us. And again, this recording will be available later on today. We will be sending out emails to everyone with the link of the recording. Once again, thanks again, and looking forward to seeing everyone in our next upcoming free webinar. Thanks, Amanda, and thank you, Eric, as well, for sharing your lunch hour and covering these important topics.
Thanks, everybody.
Thank you, everyone.
Bye-bye.
Take care. Thanks so much.